I'm Liz Wiltsey, and this is my exam mini video for Genetics 311. I chose prompt 2. In this video, I will be talking about transcription and prokaryotes in reference to initiation, elongation, and termination steps. In this first part, we see the -35 and -10 consensus sequences at the top. These allow for the core enzyme and sigma factor to bind to the promoter area so that RNA polymerase can find its way and begin initiation at the +1. In this part, we also see the transcription bubble where transcription takes place. This also shows the NTPs added that enter with the promoter to help the process of elongation. There is no primer needed in this initiation step. Another good thing to know is that the sigma factor has to be removed from the hollow enzyme so that polymerase can begin moving and initiate elongation of the sequence as a core enzyme. If it remained in the promoter, the core enzyme would not be able to move along and begin its process. In this next part of elongation, now that the polymerase has begun to move, this process can start and move the transcription bubble along the DNA strand, winding and unwinding, transcribing the RNA. Here we can see that the RNA adds 5 to 3 anti-parallel to the template strand and moves in the direction towards the right. Lastly, the termination step. This can be caused due to two different things. The first is row-independent termination. This occurs if a hairpin or inverted repeat causes the polymerase to pause and disassociate RNA from DNA with the weak AU bases. An example of an inverted repeat causing a hairpin would look like this. The second reason that termination would occur would be due to a row attaching to the growing transcript. This row would then move down the DNA transcript and catch up to the bubble where it would show helicase properties and then disassociate and disattach the DNA from the RNA, ending transcription. A 5 base pair mutation between the -35 region and the -10 box of a gene coding for an enzyme required for the production of the amino acid phenylaline can have many consequences, as this can affect the levels of mRNA due to a mutation within the promoter region. Affecting levels of mRNA can adjust the levels of protein produced or not produced in the body and in the prokaryote, affecting just about every aspect of the organism.